Getting back to our electrophysiology session, we're on session 2 now, and we're going to focus more on neuronal potentials. And these neuronal potentials are the electrical activity that we're trying to record from our electrodes in the brain. So when we sample the brain, there's many different ways of sampling it. We could do structural imaging. We could do blood flow measures. We could do radioactive particles, magnetic fluxes, and electrical potentials. And there's positive and negative things to each of these sampling methods. However, I will argue that the electrical potentials are recording from the individual neurons in the brain and not trying to see a mass activity. We can actually go down to the single neuron and see how it's involved in a particular behavior, such as moving your arm, moving your leg, how you learn to move your arm, how you learn to move your leg, and that these other measures, they do have some advantages because some of them are non-invasive. For instance, structural imaging, very, very informative to help us identify different structures in the brain, to find abnormalities. However, you can't see activities here. Within these different structures, there's multiple functions. So there's no line saying this part of the thalamus is involved in motor function and this part's involved in vision. There's no lines here. However, at the single unit electrical level, we can go in and sample these various parts of the brain and say, oh, this is a motor area, this is a non-motor area. We can look at blood flow. This is an example of what's called functional MRI. And with functional MRI, we can use various things to look at blood flow from the imaging technique. So it's pretty valuable. We've actually can identify general regions involved in arm movements. We can have the patients play tasks involved in learning and see what parts of the brain are getting more blood flow during that process. However, one of the drawbacks is it gives you a general area. And secondly, inhibitory mechanisms in the brain require energy just like excitatory. So it doesn't tell you if that area is inhibited or excited. At the single unit with electrophysiology, you can actually see if the neuron's firing more or less. So you get a concept of inhibition versus excitation. This just tells you what part of the brain is utilizing energy. We can use radioactive particles that go into the brain and bind. Here's an example of fluoro gold. And we can look at metabolism, for instance, essentially energy utilization as well. This is PET imaging. Again, a non-invasive technique. You can go in and look right after a seizure and say, is there a part of the brain that's more active than it normally is if you compare it to a non-seizure time period? So it does have value. However, it doesn't give you that individualized value. What is this part of the brain exactly doing? And is it excited? Is it inhibited? It doesn't give you that information. However, it does provide important information for certain diagnostics. We also have what is called MEG. Yet, here's an example. There's the shielding, and within the shielding are hundreds of small inductors that can measure magnetic flux. So yet, a non-invasive method, you put your head in here, you have a seizure in there, you could pay a behavioral task, looking at arm function, for instance. And what will happen is, as the part of the brain that's involved in that function will have a change in its magnetic properties, which then are captured, and we can reformat that back into the brain to know where that flux and magnetic field was. Again, not highly localized. You can't tell what the single neurons are doing. And is that flux due to inhibition or excitation? The other problem with these, especially MEG, is localizing where the activity is, because the power of the magnetic field shrinks by distance. And the closest thing to these inductors is the surface of the brain. Sometimes it's very difficult to know if it's deep in the brain or if it's the cortex of the brain. So it becomes very difficult to localize with this technique. And again, I'm going back to single units. I am biased, but I wanted to bring this concept to you about how I look at the scales of recording. And this is a very simplistic way of looking at it. So imagine that the macro contact is a really big speaker or microphone, and you place it in the middle of the cafeteria. What you're going to pick up with that microphone is the talking and the hum of everybody in the room. So what you can get a concept of is the basic hum in the room. When we go down to the local field potential, now instead of a big microphone in the middle of the room, we put a microphone between two tables. Now we can discern maybe not individual speakers at the two tables, but basically the information of those two tables, the hum of those two tables, now eliminating the whole hum of the cafeteria. And when we go down to the actual individual recording, the single unit recording, we can hear each of the individuals at the table now. And then we can move our microphone from table to table and sample individuals at the different tables, so we can understand how each of those individuals now contribute this whole harmony of the brain. And that's an important concept. We're going down and down, higher resolution in our information and down to the individual. And then we can now, for the first time with Electrode, really capture that whole domain, from what the whole room's doing to what the individuals are contributing. And we can imagine, if we just take epilepsy, maybe there's 2 epileptic cells that you can record from at an individual table. And you're like, wow, they're involved in the epilepsy. Once we make microphone bigger and bigger, our ability to see those 2 abnormal people or cells is diminished, because we get the hum of all this overriding what you can hear from the individuals. So my hope is, for epilepsy, that we're going to find out a whole bunch about how individual cells may look normal, but yet we don't see that same abnormality at macro level. And this may provide great insight on a change in how we diagnose foci. Now you have this big macro contact recording the whole room, and we're trying to find some abnormality. And it may be just washed out by the total number of people in that room. As I told you, we have these sodium-potassium pumps. It creates a charge differential. So that's called its resting potential. When we have a ligand come in and open channels, that change in current, the small potentials to move along the cell membrane. They could be excitatory, as in this case, or inhibitory. Oftentimes, inhibitory mechanisms require actually more energy than an excitatory mechanism, because they involve what are called second messengers. So the neurotransmitter binds, causes some change in a protein that then affects this effect protein. This effector protein then opens some channel. More complicated, these are called G-coupled proteins, the inhibitory. So here's an example of more energy being utilized to cause an inhibitory process than to cause an excitatory process. Both of them utilize energy. When you record local field potentials, you're primarily recording this type of activity, and it's primarily the dendritic activity that we're recording. When we do single unit physiology, we look at an action potential, we're looking at what that neuron is outputting. Whereas macro recordings are looking what's happening at the input of the neuron, not at the output, uniquely distinct types of information. So as I said, we can put neurotransmitters that cause or stimulate, which cause a depolarization of the cell. And if it depolarizes enough and that activity gets to the axon hillock, it will generate an action potential. We can also hyperpolarize the cell. That means inhibit it. So if we stimulate in a certain negative way, we can actually inhibit the cell and prevent it from firing. The neurotransmitters do both. So they're coming in, some are exciting, some are inhibiting, and that activity is traveling through the dendrites to the cell body, to the axon hillock. And it summates the positives and negatives. And if you have equal positive and negative, you get zero change in current. If you have more excitatory or depolarizing activity than hyperpolarizing, and it's sufficiently large enough, it will generate an action potential. I talked about temporal and spatial summation. So if enough information activates the axon hillock, that activity traverses the axon in the form of an action potential down to the synapses. The action potentials then cause the synapses to release drugs or neurotransmitters onto the next cells, whether excitatory or inhibitory. And then they do their summation and project to the next neurons, either an action potential or they don't fire at all. So this is the shape of an action potential, and it's an idealized shape. In fact, different cells in the brain have different shapes of their action potentials. And that's because they have different distributions of channels in. So let's just look at what's happening here. The differential in charge between the inside and the outside of the neuron is held at about negative 70 millivolts. Some neurotransmitter comes in, it opens some of the sodium channels at the on the cell membrane. And if it's sufficiently large enough of them come in and the current gets high enough, it will trigger the rest of the sodium channels to open. And then you get an all or nothing event. It will shoot all the way up. Here's your action potential. And during the different phases of your action potential, different channels are opening. So at first, the sodium channels are opening, causing a positive change in the voltage. But as all the sodium channels are opening, they reach some point where now all of them have opened and some of them start to close again. Also, at the same time, these potassium channels open around the peak, which then starts to make the charge go negative. And in fact, all the sodium channels have now closed. They're all going, the potassium's coming in, reversing the polarity. And then down here, it's hyperpolarized, which means it's below its resting potential. And this hyperpolarization is actually necessary to reset all of these sodium channels. We have the sodium channels which drive the voltage up. They shut down and the potassium then invades, and then the voltage goes down. And that hyperpolarization period makes the proteins of the sodium channels go back into their ready to open phase again. And then the resting potential's here. Interestingly enough, after this hyperpolarization period, this neuron is more likely to fire because we'll say 100% of its sodium channels are available to open. So you can actually activate a neuron right after this hyperpolarization is more likely to fire than any time period after. The whole goal of our recordings is to get clean recordings. And we can see each of these inflections is a neuron firing. And we can see even in the same recording, there's different neurons. So we have this neuron here who fires higher than this neuron here. To get this type of recording, we have to have the right type of contact close to the neuron and void of environmental artifact. We'll talk about that as we go along. But I wanted to point out that, as I had said, neurons fire differently. Their shape is different. And I told you the action potential is different. And we can see this is one part of the thalamus here, the dorsal thalamus, how it fires. This is a VIM of the thalamus. It fires differently. We can see even it has two different types of cells. It has a more sustained firing, and it has another type of neuron that kind of bursts fires. Then when we get down further in the thalamus, we get into a different type of firing pattern. So these different patterns of firing, the shapes of the neurons, all allow us to know what part of the thalamus that we are in. There's different factors that lead to the ability to record these spikes in this fashion. And if we don't meet those factors, it looks more like a field potential rather than single unit. We can't discern individuals. We certainly won't be able to see that this bursting pattern relative to this. And that comes down to the dimensions of the contacts, the impedance, and what we're actually recording from. I just have these three different things here. This is a single unit electrode used for deep brain stimulation. The tip on it's roughly 30 micron exposure. This is what we record single units for when we go in for DBS cases, and we can record those nice crisp recordings that you saw on the prior slide. However, if we go to scalp, we have these really big contacts on the surface. We're recording from thousands of units, and they're not close to them. And some of them may be deep, so it becomes harder and harder to isolate individual function. Now we go to a Dixie implant, and we can see these very, very nice electrodes with the metal contacts. Each of these dark spots is a macro contact. Now we're within the brain. We're closer to the population. We're listening to the cafeteria here, and we're listening to the individuals at the tables here. And it requires different dimensions of the contacts and different impedances. The impedances on these macros is far lower than they are on the micro. So examples, local fields are EEG, ECOG, SEEG with single unit physiology, with single unit. There's three different types. Most of what I've shown you is all extracellular activity, but there's also intracellular. Right now, I don't believe there's any way of doing intracellular in human. What we do in these experiments is we put a glass pipette inside of the neuron, and we only record from that one neuron. The patch technique, we actually put the glass pipette to the membrane of the neuron, and we put a little vacuum, and we're only recording the activity of that membrane, one neuron. Extracellular, we have neurons out in space and our contacts between them, and we're seeing the electrical flux recording from the extracellular space. The problem is, as you get lower and lower resolution, with extracellular, I can discern maybe 3 neurons in a recording. Intracellular, you're only seeing 1 neuron at a time, and with patch, you only see 1 neuron at a time. The next level of extraction is what's called multi-unit. And that's where you can see inflections, but your recording is not good enough to isolate individual neurons from those inflections. So you may have 3 neurons but the spikes all kind of look alike and you can't separate them. So this extracellular recording gives you that ability to separate. And typically what the difference is the impedance of the electrode or your ability to get close enough to the neurons, which separate true single unit extracellular from multi-unit. Here's an example of a cell attached. This is a membrane patch. You can see the cell body is being invaginated a little bit because the glass is pushed on it. This is an example of an extracellular contact. Here's an individual neuron. Here's another individual cell body of a neuron, another one, and another one. So if we look at the single unit contact coming in to do the extracellular recording, we're able to get close to this neuron. So the spike we detect from this neuron will be larger than the spike we detect from this, and much larger than the spike we could detect from this. So we might be able to discern these three neurons from our recording based on height. However, this guy might be too far away and we're not able to separate it. If you don't have a good enough electrode, a high impedance, a good enough impedance electrode, what you do is record from all three, and they'll kind of look the same. The heights are all mushed together. It's very hard to separate the individuals out. But I want to bring your attention here. I told you that the microcontact exposure, the tip, is only 30 microns in size. Neuron, or the cell body of a neuron, is about 10.8. And here's an example of our electrode. It's 15.9 micron cross section. So we're actually on the order of the size of the neuron we're recording with our electrode. One other thing to point out here, our macro contact is 2 millimeters in size. So if you were to have that on the screen, it would cover everything here in relation to this image. So imagine how many neurons surrounding a macro contact versus the fine resolution you get of a single unit contact. So what we're measuring here, these sharp inflections, are individual action potentials. The height, I said, is determined by how close you are to the neuron. Actually, the voltage drops off at 1 over r squared. In relation, magnetic fields drop off over 1 over cubed. So a small change in distance is a huge drop in magnetic field, whereas electrical fields drop off by r squared. Underlying each of these inflections is an action potential of a slightly different morphology depending on the cell. The firing pattern, cells have different firing patterns. Obviously, if let's say you're in a motor region and you move your arm, this pattern of firing will change. It may go up or down in intensity. I'm blowing up this picture to show you scale. On the bottom here, we have 2¼ second recording. And you can see this neuron's firing quite fast. Neurons can fire 40, 50, 60 spikes a second down the 2 to 3. If I blow up just a 1/4 second, we can see here, I was able to separate this spike, which is labeled green. Here it is again firing, labeled green, from the other spike in the recording, which is this yellow one labeled yellow. And what we can see is they kind of look the same, the yellow ones, and the green ones kind of look the same. And that's because when they fire an action potential, it's a very consistent, reproducible electrical flux. So spikes. So I don't sit here and blow up a 1/4 second and then say this is neuron 1 and this is neuron 2. We have very sophisticated software to help do this. So down beneath here, we can see a trace and we can see there's spiking activity. It's not the best picture, but then we have a computer algorithm that can separate it into individual neurons. In this case, it separated this recording into 1, 2, 3, 4, 5 different types of neurons. We do go back and check to make sure it's a reasonable way of separating the neurons. Now, you can imagine when I started doing physiology, we recorded 1 electrode at a time. With this new MME we have, we can record, depending on the model, up to 12 recordings at a time. There are other types of electrodes where you can record hundreds of single channels at a time. So we need computers to help us. There's no way we can monitor all 100 channels. How do we sort 100 channels? We'd be in the lab just doing that all the time instead of the analysis. So we've developed algorithms to help us go through this process. I'm not going to get too much into data analysis. What I will do is show in later segments some of the type of studies that you can do with single unit. And that pretty much is the end of this session.